Hello, it's Mr. Cho here, and actually today you'll learn why my surname is so important. So, in today's chapter, we are going to learn about biological molecules. Okay, so this chapter is divided into two parts. We will firstly learn about various nutrients, and then we will learn about this thing called enzymes. Okay, let's look at this picture. So here we see people running a marathon. Usually a marathon is either 10 km or 42 km, right? So it can be a very long race, and if you are planning to win the marathon, then you need to really work hard for it. Now, do you know that marathon runners usually need to eat a lot of carbohydrates 2 days before a race? Now, this is known as carbo loading. Basically, you load your body with carbohydrates. Why do they do that? Even though we know that eating too much sugary foods or food that is high in carbohydrates can lead to obesity or even things like diabetes. So why do they eat that? That's because there's a function for this nutrient, right? And the function of carbohydrates is as a source of energy. Okay, so what are carbohydrates? Learning outcomes list the chemical elements that make up carbohydrates, state that cellulose, glycogen, and starch are synthesized from glucose. State the main role of carbohydrates as an immediate source of energy in living organisms. Describe and carry out tests for starch using iodine in potassium iodide solution and reducing sugars using Benedict solution. Okay, so all living organisms need food. The food that we eat contain nutrients that are essential for us. Nutrients include water, carbohydrate, fats, and proteins. Carbohydrate, water, and proteins are the three major types of biological molecules found in living organisms. Water is an essential component of all body tissues. About 70% of our body weight is water. Hence, water is extremely important to life. Okay, so here in biology textbook, you have these three main nutrients, and water also actually four. There are actually some other nutrients such as vitamins and mineral salts that you might have learned in maybe food and nutrition science, but we are not really focusing on those in the biology textbook. Okay, so in the old syllabus, right, we actually learn about the functions of water. So just a quick note on this, right? Water has several functions, right? It dissolves substances that allows for chemical reactions to take place in living things. It's essential for photosynthesis, keeps the plant cells turgid, and helps to transport mineral salts. In humans, it regulates the body temperature and helps to transport dissolved substances in the body. Okay, so water is a very important part of our body. So let's now talk about carbohydrates. Carbohydrates are organic molecules made up of the elements carbon, hydrogen, and oxygen. The hydrogen and oxygen atoms are present in the ratio 2:3. Okay, so the word organic, it means containing carbon. So we humans are organic matter, right? Because we actually we contain carbon in our body. I was curious, so I went to find out what are humans made up of. We are actually made up of these elements, right? We are made from oxygen, carbon, hydrogen, nitrogen, and others, right? Such as iron, calcium, etc., etc. Okay, so there's one over here. Right? All of these are the elements that make up a human, right? So we need all these elements to give us life. And just like human, right, it's made up from carbon, hydrogen, oxygen. The carbohydrates are also made up from these same elements. Now, a glucose molecule has a formula of C6H12O6. That is to say that there are 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen to make one molecule of glucose. Then a sucrose molecule has the formula C12H22O11. Alright, so a sucrose molecule is what we call as a double sugar. Okay, we'll learn about that later, about this thing called single sugar, double sugar, and complex sugars. Okay, so that's why it has twice the number of carbons as compared to glucose. So glucose is a single sugar, there's only 6 atoms of carbon, whereas for sucrose there are 12 atoms of carbon. Alright, yes, over here. So carbohydrates can be divided into two major groups: sugars and complex carbohydrates. So when we talk about drinking sugar, right, most of the time they are referring to things like glucose, fructose, maltose, and sucrose. Okay, so glucose, this is a form of sugar, right? It is what our body uses to generate energy. So most of the time, whatever we eat, right, it most likely ends up as glucose, right? So for example, the starch that we eat, it will be converted or broken down into glucose, which our body can then use. Other things like fructose, our body can also use it, but they will do certain conversions to it to make it become glucose in the end. Okay, fructose is a kind of sugar that is found in sweet drinks. For example, your Coca-Cola, your 7-Up, all these all contain fructose. Okay, let's take a quick look at that. Coca-Cola fructose. Okay, so yeah, Coca-Cola is made up of about 60% fructose and 40% sucrose. Probably it depends on the formula that is used to make Coca-Cola. Okay, so as you can see here, yeah, one of the main ingredients in Coke is high fructose corn syrup. Okay, so actually, fructose is a very sweet tasting sugar. Glucose also is quite sweet, but it doesn't taste as sweet to our taste buds as fructose. Okay, now we talk about double sugars. Maltose. Okay, so where do you find maltose? Usually in products that are made from malts. Right, so maltose. Maltose is made up of two glucose molecules. That's why it's called as a double sugar. Okay, so maltose, let's see, products. Okay, one common drink that we actually drink in Singapore is Milo. So Milo contains malt. There is maltose in Milo. Okay, then sucrose. Okay, sucrose also is a double sugar similar to maltose. Okay, so just like similar how to maltose is, except the sucrose is made from a glucose and a fructose molecule. Okay, you don't really have to remember all of this. Why is there a Genshin girl here? That's weird. Her name is Sucrose. What? Okay. Sucrose food products. Okay, so yeah, when we eat things like chocolate, maple syrup, or sugar, these are food that is also high in sucrose. Okay, I want you to put a star on the word sucrose and write down non-reducing. Okay, non reducing. Okay, so we will talk a little bit about food tests later, about how we can test for this thing known as reducing sugars. Reducing sugars will do something to a food test. Okay, but sucrose, being a weird person, that Genshin girl or something, is the only one that doesn't react in this food test. Okay, so put a star on this sucrose and write non-reducing. Okay, now let's talk about complex carbohydrates. So this is a bit easier. Complex carbohydrates are what we find in these three things: starch, cellulose, and glycogen. Okay, so what is starch? Starch is a long chain of glucose molecules. Where do we find it? We can find it in various foods such as bread, noodles, and rice. Alright, all of these are made from starch. So let's take a look at starch. Starch. Okay, so starch is basically a chain of glucose molecules. Okay, imagine that each of these circles represent one glucose molecule. So a long chain, right? Usually it's actually much longer. It's like in the thousands, thousands of glucose, okay, something like this. Okay, thousands of glucose molecules that are combined together form the starch. Okay, so it can be represented by anything, maybe this shape or circles or even squares. But as long as you see the same pattern of the shape, then you know that it's glucose molecules that are combined together. Okay, so that is starch. Then cellulose. Okay, cellulose is what is used to make the cell wall. Okay, so let's take a quick look at that. Cellulose. Okay, so yeah, like if you eat vegetables, I mean you should eat vegetables, right? The cell walls are made up from cellulose. And just like similar how starch is made up of glucose, cellulose is also made up of glucose. Okay, so over here you can see it's just that they arrange the glucose in more orderly ways. So as compared to starch, where it's a bit like a spider web looking, cellulose tends to look the glucose is organized more neatly, something like this. Okay, more neatly and stacked up in an orderly way, so that it provides a strong cell structure. You'll provide a strong structure for the cell wall. Okay, so this is how it looks like. Okay, so then glycogen. Okay, glycogen is just like the previous two. Glycogen is also a storage form of glucose, but not in plants. It is found in animals instead. Okay, so in plants, right, the glucose molecule is stored, that combined together in either starch or as cellulose, but in animals, the glucose molecules are combined together in this thing known as glycogen. Okay, where is glycogen found? It's mainly found inside our liver as well as in our muscles, right? So it's basically a way of our body's way of storing energy. When we need energy, right, we can use the glycogen, convert it back into glucose, and when we want to conserve energy or save energy, we can actually convert glucose into glycogen, and back. It's like a back and forth thing. Okay, so glycogen, as you can see, is also very a long chain of glucose molecules that are all combined together. Okay, so wow, I talked so much, but actually there was a lot of things here. Okay, come repeat myself. Okay, so sugars, right? Sugars are sweet and soluble in water. When they dissolve in water, they will lower the water potential of the solution. Sugars provide us with energy to do work and carry out activities. Okay, so here you can see to exercising. When you exercise, you are mainly using the simple sugars in your body, things like glucose, right? Because that's what our body uses for aerobic respiration. Okay, so here a single sugar is the basic unit of a carbohydrate. It can pass through cell membranes and be absorbed into the cells. Okay, and this is why digestion is important. If we don't digest the food into single units, right, the basic units, then it cannot be absorbed into the cell. So our common single sugars are glucose and fructose. They have the same chemical formula, C6H12O6, but the atoms are arranged slightly differently from each other. And yeah, where do we find? Okay, glucose is commonly found in plants and in animals, whereas fructose is common in plants but rarer in animals. Okay, rare but still possible. And there are actually two others, right, called galactose and something that I can't remember, but those are lactose, but those are not important at all. Okay, so you can just ignore those. Okay, now let's talk about this thing. Okay, so yeah, glucose is found in plants and animals. Fructose is common in plants but rare in animals. The structure of glucose and fructose can be represented by the diagrams shown in figure 3.4. Okay, so how do I read these kind of diagrams in chemistry? Okay, it may be useful for you to know in the future. Basically, each of these points, each edge, right, is referring to one carbon. So you can count 1, 2, 3, 4, 5, 6. So there's a total of 6 carbons. Okay, they are not showing you the other things like the oxygen and hydrogen, because the most important thing in the molecule is actually the number of carbons. Same thing for fructose. You can count the number of edges. 1, 2, 3, 4. 1, 2, 3, 4, 5. Okay, this is arranged a bit weird. Let's see. Fructose molecule. Okay, yes, I forgot, because fructose have a carbon that's hanging on the right. Okay, so fructose, right, it'll be 1, 2, 3, 4, 5 carbon molecules, and then 1 carbon molecule that's just dangling at the side also. Okay, so then we have our double sugars. Okay, so double sugars are formed when 2 single sugars are joined together. The 2 most common double sugars are maltose and sucrose. Maltose is known as malt sugar. It occurs in germinating grains. Maltose consists of 2 glucose molecules bonded together. Right, so here, 2 glucose, bam, smack together, form maltose. This is a chemical reaction. Then a sucrose molecule is made out of 1 glucose molecule and 1 fructose molecule joined together. Glucose plus fructose equals sucrose. Okay, and you may have learned this in Sec 2 science about the enzyme called maltase. Okay, so remember, enzymes always end with the word ase, ACE. So what happens here? When you have a maltose molecule, you can be split or digested or broken up using the enzyme maltase into 2 single glucose. Okay, and this is the end point of digestion. This glucose can then enter into the the cell membrane, right, and then can be used inside the cell. Alright, now let's talk about reducing sugars. Okay, so just now I asked you to label down the word sucrose, right, and say that it's a non-reducing sugar. Why? Because this is one of the basic food tests. Okay, what are food tests? Food tests are basically a way of testing whether an unknown substance is present inside your food. Okay, in this case, we are going to test for reducing sugars. Now, reducing sugars are 3 things: glucose, fructose, and maltose. These are reducing sugars. Reducing sugars will produce a red precipitate when boiled with Benedict solution. Sucrose is a non-reducing sugar and will not give a red precipitate with this test. Okay, why is sucrose so special? Is because it is a little bit different. It behaves a bit differently in its chemical makeup. Okay, so it doesn't do this thing called oxidation. Okay, so anyway, just ignore it. Just know that sucrose is non-reducing. Alright, this is a very important test which all of us must know how to do, right? Whether pure bio, combined bio, NA bio, everybody need to know how to do this test, right? Because this one is commonly very common question, both in theory and in practicals. Okay, so the aim of this test is to test for the presence of reducing sugars in different food samples using Benedict solution. Okay, what is Benedict solution? It is named after a guy called Benedict who invented this solution, and this solution is basically it's a blue color solution. Okay, let's just take a quick look. Benedict solution. Okay, every time I type Benedict, there is the Benedict Cumberbatch come out. Okay, so Benedict solution is on its own, it is a blue color solution. Okay, so let's see what happens to the Benedict solution when you do different things, when you put it in different things inside. Okay, so firstly, how do you do it? You will take 2 cm cube. Okay, 2 cm cube is another way of saying 2 ml, which is roughly the volume of this area. Okay, so if you take your finger, right, the first the first section of your finger, that's roughly about 2 cm cube. Okay, if you don't have a ruler or what, you can just use your finger to estimate. About everybody's finger is slightly different. Okay, so put 2 cm cube of the food sample into the test tube. Then add 2 cm cube of Benedict solution to the food sample. Okay, so you can see here, right? That's the food. Then you add the Benedict solution. Shake the mixture. Okay, why do you shake the mixture? It's so that you can evenly mix the contents, right? The food and then the Benedict solution should be evenly mixed inside. And then you place the test tube into boiling water bath for only 2 to 3 minutes over here. Okay, so you should make sure that the water is boiling already before you put the test tube in. Not you put the test tube in, then you wait for the water to boil. Okay, the water must be boiling at the moment you put it in. Okay, Take note, you should only heat it for 2 to 3 minutes. Any longer than that can actually result in what you call as a false positive. The meaning of false positive means that you get the result as positive, but it was not really supposed to be positive. Okay, there's a reason why. For example, if I'm, let's say, testing starch, right? Starch is not a reducing sugar. Starch is not going to turn any color, right? It's going to, actually, it should be, the solution should remain blue in starch, but because of the high heat, right, the heat can actually cause the starch to break up, right, based on the heat, and there'll be small little bits of reducing sugar such as glucose that then floats out from the starch, and that can cause the test to become positive. Okay, so let's look at what are there are 4 colors that we need to remember for this test. Alright, so the first thing is, the first observation is that if the solution remains blue, that means that there's no reducing sugar in the food. Okay, so for example, if I put like some protein or some oil or whatever that doesn't contain any reducing sugar, and I mix it with my Benedict solution, I heat it up, I should still see that the solution remains blue. Okay, so just remember, the word remain is also important to write. You can't just say solution is blue, right? The solution is remaining blue. Then, if I see that the Benedict solution turns from blue to green, okay, that means that there's a trace amount of reducing sugar. Okay, actually, I feel that the observation should be written in another way. Okay, how I would write the observation is that the Benedict solution turn from blue to a green precipitate. Okay, and I'm gonna explain later why we write this word precipitate. Okay, so but for now, can we just put like a star here, from blue to green precipitate. Okay, the spelling for precipitate is over here. Okay, so next, okay, the word trace means very little, like very small amounts of reducing sugar. So if you see this, that means that yes, there's a bit of reducing sugar such as glucose, but just a very small amount. Next, if the Benedict solution turn from blue to yellow or orange, again, put the word precipitate at the back. Okay, so Benedict solution turn from blue to yellow orange precipitate, then this is what I call as a moderate amount of reducing sugar. The word moderate means that there's, there is some significant amount, but not a lot. So moderate. Lastly, the observation is, if the Benedict solution turn from blue to a red precipitate form, this is the best. Okay, this means that there's a large amount of reducing sugar present. Okay, so some teachers will actually ask you to write the word brick red. Okay, so it's not just any red. This is actually called brick red color. Like, so it's the color of bricks. Yeah, it's just something that I learned when I was in school. Okay. So let's watch a quick video on this test, Benedict's test. Oops, no internet. Just give me a moment. Okay, so let's watch a video on the Benedict's test.
Add about 2 cm³ of glucose solution. We can use a dropper to measure exactly 2 cm³ into the test tube first.
Okay, in case you didn't catch that, the dropper, there's lines on the dropper, so when you suck up the glucose or whatever food sample you're sucking, you try to make sure that it sucks up until the line that says 2.
Into the test tube first. This is followed by adding an equal volume of Benedict solution, and you notice that we will change another dropper so as not to contaminate the solution. Again, we use a dropper to measure exactly 2 cm cube of Benedict's solution and add it to the test tube. Shake the test tube to mix the contents. Then we will heat the test tube in a beaker of boiling water. First, we use the test tube holder to secure the test tube. Then we put the test tube into the beaker of boiling water. And we make sure that the water in the beaker is higher than the solution in the test tube.
Okay, so why do we do that? This is so that all the contents will be evenly heated. If the water level is too low, like let's say below the line of the test tube, then only the bottom part is being heated and it's not heated evenly. The solution will have to move by itself to heat. It will heat up eventually, but it's not even.
We will leave the test tube in the beaker of boiling water for 3 to 5 minutes.
Okay, this is an old syllabus. So this one, not really 3 to 5 minutes, it's actually 2 to 3 minutes. And if it's too long, it doesn't look good also, right? The color will start to become very funny colors.
Observe the color change. The original color now is blue.
Okay, so you can start to see that there's a green precipitate forming at the top, right? It's not always immediately obvious, and you also don't need to go and use your finger and stir this thing or use your thing to stir. Just let it move by itself. Okay, same thing, right? From green now forming a yellow precipitate. Then we should see. Okay, so sometimes it's yellow, sometimes it's orange, depending on how much reducing sugar there is. But if there is a large amount of reducing sugar, the final product, the final color that you see will be what we call as a brick red precipitate. The brick red precipitate, some people will say that it's actually brown, or to them it looks brown. Yes, so if you see it as brown, that is a brick red precipitate. Okay, so this is the color of the brick red precipitate.
Once it turns into a brick red precipitate, we will off the Bunsen burner. And now you can see the orange red precipitate.
Okay, so why is it that I keep calling it precipitate and not solution? So when we do a Benedict test actually is creating that the products, right? They are actually creating this small little fine little powders inside the solution. Okay, so a solution means that it's clear. You can see through it. That's the meaning of solution. But precipitate, right? See here, green precipitate, orange precipitate, red precipitate. It means that it's actually very fine little like dots or little specks of dust that are green in color or orange in color or red in color. If you actually let it rest for a long period of time, the color actually can go down. Okay, so that's why it's called precipitate. Okay, the meaning of the word precipitate. What is precipitate chemistry? Okay, so this is the meaning of the precipitate lah. Okay, there are all these little things here. Okay, so you can see more clearly that these are precipitates, right? It's similar to, like, remember you learned about your solution and suspension, right? There's all that little particles that are floating around in the suspension, and then they can precipitate together. So a precipitate is actually a solid product. Okay, so a precipitation reaction occurs when dissolved substances react and form a solid product. Okay, that's why we have to be a bit more specific. And use the word precipitate and not the word color, right? When we're writing for this part on reducing sugars using Benedict solution. Okay, all right. So let's quickly let's just pause here. Alright, and we'll continue the chapter in the next part of the lesson. Okay, see ya, peace out.